Okay, now that we are recording, do some reminders. Of course, I'm probably preaching to the choir because if you're here right now, you probably don't need reminded of anything. But for the people watching the video, maybe you could do some reminding. So lab, first reminders about lab. This week's lab is due tonight. Remember, there's two parts to it. There's that virtual experiment that you go through, and then you take a screenshot when you're done to prove that you did it, and then you watch that video while answering questions. It's pretty simple, but I guess it would be, easy to overlook it. People overlook things all the time, so make sure you do both parts. So that's this week's lab, and then next week's lab, I'll remind you that the pre-lab is due this coming Sunday. It's an easy one. All you need to do basically is watch a short video, one of those crash course biology videos. I think it's like 13 minutes long, and answer some questions to prove you did it. So don't forget to do that by Sunday. And it will be an online lab, right? You're going to you're going to do the experiment online. So you can go ahead and do it. You don't need to wait until Wednesday. But if, for those of you who don't do it by Wednesday and you haven't heard anything else from me, let's plan to meet here on Wednesday at noon as usual. So yes, plan to be here unless you hear otherwise from me. If you want to hear otherwise from me, your best bet is to do the lab early. And also, don't let me know, or let me know if you need help with it. So just because lab technically isn't until Wednesday, that doesn't mean I won't help you do it early if you want to do it. So let me know. I think that's it for reminders. Any questions about anything? All right. Let me update my attendance really quickly. There are such a small amount of people here, and even online, I'm giving you guys just a little bit extra credit just for physically being here. So 2.5 instead of 2.4. And good job on getting that extra credit yesterday. It's good to know. I know you were one of the people that had a screen in front of you, but apparently you were still paying attention. Anyway, no questions at all? Okay, let's jump into chapter 10. The structure and function of DNA. In my opinion, and also based on what I've observed from students past, this is not the easiest chapter of the semester, but I think it is the easiest of the last three chapters. So the last three chapters, total three chapters. So chapters 8, chapter 9, chapter 10. That's going to comprise of exam number 3, right? So we are now in the last chapter of the third exam. Oh yeah, sorry. Attendance words. The first word will be exam. Since we're talking about exams, that's the first word for attendance. Make sure you send in your keywords online. Make sure you send them in by 9 a.m. Anyway, so this is the third chapter of the third exam, and I think it's a little bit easier than the other two. It's definitely shorter, and since we're talking about the structure and function of DNA, some of this will be a review, because if you remember, we learned about nucleic acids when we did chapter 3 and learned about biological molecules. And I said a lot of this stuff, I said, all right, I'm going to go through this quickly because you're going to learn it again later in the semester. So here we are now, later in the semester. Any questions before we jump into it? All right. Let me pull it up. Oh, it's not working for me. It's a Friday. Here we go. Why molecular biology matters? I should say matters. It says matter. So first of all, I haven't even introduced you to that term yet, molecular biology. But basically, when we're studying DNA and RNA and all those NAs, right, all those nucleic acids, basically we're talking about molecular biology. In a sense, and this isn't completely accurate, but you could almost think of it like we're still looking at the stuff we've already learned, like heredity, right, all the chromosomes and all that. We're kind of still looking at that, but now we're looking at it at the molecular level, literally. That's why it gets its name, molecular biology. So we're no longer looking at the result, like, all right, if you cross this person with this person, this is the results, right? We're not looking at that. We're zooming in. We're actually looking at the molecules of nucleic acids. Anyway, let's take a look at this picture. First of all, I have to remind myself what this picture even means. Yeah, there's no way you can guess that. Basically, what your book says, the caption for that picture right there on the top left of this person looking at some liquids, it says all life on Earth shares a universal genetic code, so your DNA can be used to genetically modify bacteria and vice versa. So that's what's going on in this picture. I could assume, and I don't know, but if we're talking about genetically modified organisms and we're talking about bacteria and humans and vice versa, One good example of that is insulin. Have you guys ever heard of insulin? Right, so you know insulin, unless you have diabetes, you make a good amount of it normally in your pancreas. But people who have diabetes sometimes have to buy insulin. Where does that insulin come from? It comes from bacteria. How do we do that? Well, we took the genes from a human being that says, hey, this is how you make insulin. We took those, we cut them out of the human genome, and then inserted them into a bacteria genome. And the way it works, because it's a universal code, the bacteria doesn't know any better. It reads the gene. It says, all right, I'm going to make this protein, even though bacteria don't normally do that. And that's how we get our insulin. It used to be from things like, we used to use pig pancreas and things like that. But yeah, that's how it works now. So by the end of the chapter, you can learn a little bit more about GMOs. And I think that's really important because there's a lot of scare tactics going around about GMOs. I think people don't quite understand them, and that's why they're afraid of them. What else? Let's see. Oh yeah, how many of you have ever heard of mad cow disease? Two of you, right? Heard of it? You haven't heard of it? It used to be a lot bigger. I think back in the early 90s, maybe there was a big outbreak. Anyway, mad cow disease is a disease that you're going to learn about a little bit later in this chapter. It involves the brain. It involves misfolded proteins. You'll learn all about it when we get there. Does anybody recognize what this is? Either one of these? Good. Red blood cells specifically. That's a normal one. Any idea what's wrong with this one?
Sickle cell.
Yeah, that's sickle cell. And we learned a little bit about sickle cell in the last chapter. I told you, don't worry about it for this chapter. We're going to learn a little bit more about it in the next chapter. So here we are. We're going to learn about it in this chapter. We're also going to learn about it in the next chapter. So it gets 3 chapters. But anyway, yeah, that's sickle cell. And what causes that, as we kind of hinted at in the last chapter, is one letter. So you have a whole gene, all these A's and T's and C's and G's, all these codes, right? And you just change one letter of that whole entire gene, and because of that, you get this misshaped blood cell. Well, first of all, you get a misshaped protein, which then leads to this misshaped blood cell, which then leads to a bunch of symptoms, which we learned last chapter as pleiotropy. So one gene, many characters. Anyway, that's just that. Are there any questions about what's on the slide here? That's just the introduction. Obviously, so is this. This is just the introduction as we go through every chapter. Biology and society, I'm going to skip it again. As always, I recommend reading it. As always, there'll be no questions about this on the exam. You can tell this is an old book. I bet the new version of this book, when it comes out, instead of talking about Zika, we'll talk about COVID. Which is a good time to say, this is the most helpful chapter when it comes to understanding viruses and COVID and all that. I'm not specifically going to teach you about COVID here. I'm not going to specifically teach you about how the vaccine works, but you're going to have enough background information to where you could go out and understand how COVID works and how the vaccine works. It's actually really important, and I was just thinking about this last night because I know this sounds off, but I noticed one of my old Facebook acquaintances had blocked me. Like, I don't know for how long. I just happened to notice because his wife showed up on my news feed and she had tagged him in something, and his name wasn't a link. It was like, yeah. I've been blocked. But anyway, that brought me to thinking, back when COVID was really first hitting, there's a lot of misinformation around, and I thought I could at least correct people. And I was polite about it. I wasn't, you idiot, right? I wasn't rude about it. But I would say, hey, actually, that's not true. RNA works this way. DNA works that way. Anyway, what happens is people get misinformation. And luckily, hopefully, and this is the purpose of a college education, with enough background information, you'll be harder to fool. Right? You're not going to be experts on COVID, on viruses or vaccines after this chapter. But hopefully you'll have enough information where if someone says some of the ridiculous things that I've seen, you'll be like, oh, no, that doesn't sound right. I remember that doesn't sound right. And then you'll know where to look. So anyway, get off my soapbox. This chapter is broken down into 3 main bullet points. First, we're going to talk about DNA structure and replication. This is going to be really quick and easy because we already talked about the DNA structure in chapter 3, biological molecules. We already sort of talked about DNA replication when we talked about mitosis, right? S phase and interphase. After that, we're going to talk about how we actually go from DNA to RNA to protein. Because like we've learned in the last chapter, genes are instructions on how to make proteins. And the genes are in the DNA. So this bullet point is going to teach you how that works. And then once we know all that, then we can understand how viruses and other non-cellular infectious agents work. Any questions so far? All right. So the last chapter introduced you to heredity. and kind of did it like in a story way, right? They told you about Gregor Mendel and what he did. And your book does the same thing. I like it for this chapter. So instead of just saying, here's what we know about DNA, here's what we know about RNA, here's what we know about molecular biology, it kind of gives you the time frame. Like, here's what we used to know, and then we learned this, and then we learned this, and then we learned this, and I think it's a great way to present it. I will say this, though. For the exam, you don't need to know any of these dates. You don't need to know the steps in which we learn these things. It's just a good way of presenting the information. The second word for attendance, let's see. I will just circle it instead of say it. Second word for attendance is right there. I'll even point an arrow at it. There we go. Anyway, in the late 1800s, scientists did know that DNA was a part of cells. That's it. They had no idea what it was. They had no idea what its structure was. They just knew if you were to look into a cell, they couldn't really look into it and see it necessarily, but they just knew, and you can look that up for independent work. How do they know DNA was in cells in the 1800s? But they did. They knew DNA was a part of cells, along with some other stuff that they knew about, right? That's what we knew way back when. Then, in the 1930s, way after Gregor Mendel, by the way, they figured out that there was one kind of molecule that was the basis of inheritance. So all that stuff you learned in the last chapter, all that stuff that Mendel learned or figured out, in the 30s, we figured out there's some kind of molecule responsible for that. They didn't know what yet, but they knew there was some kind of molecule responsible for that. That's also a good independent work topic if you want to look into it. Who was it that figured this out, right? Or was it multiple people? When exactly did they figure it out? When in the 30s? But yeah, in the 30s, we knew that there was a certain kind of molecule that was the basis of inheritance. It was the basis of everything you just learned in the last chapter. Then in the 1940s, they figured out that chromosomes were made up of two components. And you already learned this earlier, and we even talked about it again briefly in the last chapter. The two components that chromosomes are made up of, as you already know, are DNA and protein. So you've seen this picture before. I mean, they didn't know this. Maybe I shouldn't even put this picture on the slide, because at this point, they didn't know this. They didn't know that's how it was structured, and the DNA wrapped around those histones and make nucleosomes, and those things would coil up. They didn't know any of that. But they did know, at this point in the 40s, that chromosomes were made up of DNA and protein. So also think, too, this is all additive, right? So at this point in the 1940s, they knew DNA was made up of two components. They knew that there was one kind of molecule that was the basis of inheritance. They hadn't put any of that together yet. Any questions so far? All right, the next one is a big decade as far as this conversation is concerned. In the 1950s, they figured out that DNA was the molecule that acts as the hereditary material. So they put all the puzzle pieces together and they figured it out. And that ushered in what we call molecular biology, which is the study of heredity at the molecular level. So we're still talking about the last chapter, chapter 9, in a way. Now we're just zooming in, like really what's happening when we're looking at the molecules. Those things that you learned about in chapters 2 and 3, right? Carbon bonds 4 times and oxygen bonds 2 times and all those things that you learned. So yeah, that was a big one in the 1950s. And I would say look that up. That would be another good independent work topic, what happened in the 50s. So we're actually going to talk about what happened in the 50s because it was pretty darn important. Yeah, any questions? All right, so once they figured that out, the next step was to figure out DNA structure, right? So they knew that DNA was a hereditary material. But as we learned in chapter 1, and as it keeps popping up throughout the semester, there's this relationship between structure and function. So it would be helpful to figure out the structure of DNA because you can learn much more about its function and how it works if you knew how it was shaped. At this point, they knew all of its atoms and how they were bonded, right? So they knew how many carbons there were, how many nitrogens, how many hydrogens, how many oxygens. They knew that, some of these carbons were double bonded to this and single bonded to that. They knew all that. But what they hadn't figured out was the 3-dimensional arrangement of atoms. Remember, that's very important. Structure and function. Structure and function. Very, very important. Especially in this case, and you'll see why when we get to it. Like, oh yeah, hopefully it'll be a eureka moment. You'll see, yes, I can see how once they figured out that structure, that the function just became immediately clear. I hope that comes to you. So again, yes, the 3D arrangement wasn't known. And again, like I said, that gives it its unique properties, so that's why it was important. Specifically, I didn't mention this. Why is it important? What they didn't know was how the genetic information was stored, right? They knew DNA did this, but they didn't know how the genetic information was stored. They didn't know how the genetic information was copied, and they didn't know how it was passed along from generation to generation. They just knew all these things happened, right? They knew it was stored, they knew it was copied, they knew it was passed along. They just didn't know how. Any questions? All right. Now we're getting into the more important stuff as far as the exam is concerned. So we're kind of done with the introduction. Now we're getting into the information you need to know. The first thing we're going to talk about or review from Chapter 3 is DNA structure. As you already know from Chapter 3, DNA and RNA are nucleic acids. Memorize that fact first, I guess. If you don't know that, then the rest of the information doesn't matter. It's not a protein, it's not a carbohydrate, it's not a lipid, right? DNA and RNA are nucleic acids. How can you remember that? Well, that's what the NA stands for, nucleic acids. As you already know from Chapter 3, they are the polymers of these monomers called nucleotides. So the monomer, I'm going to underline their boxes together because that kind of goes together. The monomers of RNA and DNA are called nucleotides, as a reminder. Now, I almost definitely ask you that again because I've already taught it, already learned it once. You're learning it again, so you really should know it by now. That's what the monomer is called. What's the polymer called? Lucky for you, this is one of those that actually makes sense. The name makes sense. The monomer is nucleotide. The polymer is polynucleotide. Right? It's right there in the name. Very simple. It can be very long. You can look that up for independent work. What's the average length? Trick question, actually. I'll go ahead and tell you, but it might be hard to find it because it depends on the chromosome you're talking about or the specific organism you're talking about. Like different species have different lengths of chromosomes. If we're talking about DNA, and I should write this down. Whoops, it should be on there. That last bullet point is in reference to DNA. It can have any sequence of the 4 types of nucleotides, which are A, C, T, or G. If we were talking about RNA, it wouldn't be a T, it would be a U. And we'll come back to that. So no need to write that down right now. It's just that technically the information on that board is misleading and incorrect. And now I've corrected it. I feel better. Yep, so that's the code, right? That's the letter. Those are the 4 types of molecules, 4 monomers that make up nucleotides. A's, C's, T's, and G's. Just like with sugar, right, the carbohydrates, there was different types of sugars that you could build. There was glucose and fructose, right? You put those together and make sucrose. Or there was glucose and galactose, right? All these different types of monomers. Here it's the same thing. It's 4 different types of monomers. Or even better, remember when we learned about proteins, there was 20 different amino acids. Like you could have this one or 19 others. And this one or 19 others, right? Here, there's only 4. It's an A, a C, a T, or a U if you're talking about RNA, and a G. That'll be very important a little bit later. But you definitely need to know it for now, right? Know that the A's, the C's, the T's, and the G's. Any questions so far? The next slide is going to show you a picture of DNA. So on the left, we have this double helix of DNA. When I say double helix, and we're going to talk a little bit more about that soon, and we've already talked about it in Chapter 3. What I mean is technically there's 2 different strands of DNA, right? One there and one there. There's 2 different strands. They're held together and they're twisted. That's what double helix means. And I'll remind you of all that later. Here, we're kind of zooming into just one of those strands, right? So these represent the same thing. We're just looking at it differently, especially these 2. These 2 do show the same thing, a single strand of DNA. They just show them in a different way. So there you have T's. A single nucleotide, we zoom in, that's a T, that's a T. They're the same thing, they're just represented different. Yeah, we'll go back to that. We'll do some more details later. But are there any questions about that so far? All right, again, talking more about this. The nucleotides are joined by covalent bonds. which is where the sugar of one nucleotide and the phosphate or another are brought together. So here's the sugar, here's the sugar, phosphate, sugar, phosphate, sugar, phosphate. The thing that keeps a single strand of DNA, or excuse me, nucleic acids together, the nucleotides, are covalent bonds. Please know that. That's very important. So I can tell you this. Let's see if anybody remembers it from chapter 2. There was 2 different bonds. Well, we talked about 3 different bonds. And of the 3, one of them was the strongest, and another one was the weakest. Does anybody remember which covalent bonds are? Or are they the strongest or they're the weakest? Good, strongest. And that's not trivial. That'll be important a little bit later on. So yes, the thing that holds this single strand of DNA together, all these nucleotides, those are covalent bonds. The strongest of the 3 bonds. All right, let's zoom into these nucleotides. Again, like we did in chapter 3. You should already know this. I'm just reminding you. The 3 parts of the nucleotides are made up of nitrogenous base, a sugar, and a phosphate group. Just like I said in chapter 3, you could just remember base, sugar, and phosphate. That's good enough, because on the exam, I'm not going to try to trick you. Like, is it a nitrogenous base or a phosphorus base? Nothing like that, right? Base, sugar, phosphate. Also, it doesn't need to be in that order. That just happens to be in the order that happens to be the order in which I wrote it. And that's what we learned in chapter 3. If I remember correctly, what we didn't do was go into detail about the nitrogenous base, and then go into detail about the sugar, and then go into detail about the phosphate group, and that's what we're about to do. So the next word for attendance will be sugar. How sweet. Are you guys ready for Halloween next week? Any questions so far? All right, let's zoom in and look at the nitrogenous base. These are the things that are projected from the backbone, meaning, and I guess the picture's not really there, but remember there's the sugar phosphate backbone, right? Those are like, obviously the sugar attached to the phosphate, attached to the sugar, attached to the phosphate. That's the long string. This part just kind of sticks out. So technically, it's attached to the sugar, but it also, the other side of it isn't attached to anything, right? So that's not going to be a test question, but that's important to understand for later. So I won't X through this. I put a little check next to it, like, hey, that's important, but you're not going to get tested on it. This part definitely won't be on the exam, so I'm going to put an X on it. And really, as far as understanding what I'm about to teach you, it's also a little bit less important. But the nitrogenous base has a ring that's made up of nitrogen and carbon. That's what that's made up of. And of course, some things off the ring, like you have some oxygens, some hydrogens, a methyl group, not that you need to know what that is. But the ring itself is made up of nitrogen and carbon. And it is basic as opposed to acidic. And that's where it gets its name, nitrogenous base. But again, that's not important for you guys. You guys can completely understand the information I'm about to give you if you don't even know those 2 bullet points. So not very important. What is important, like I've already mentioned, is 4 varieties, right? We're talking about DNA. The 4 varieties, the 4 versions of those are A, T, C, and G. This is also a good time to point out it is the nitrogenous base that defines the whole nucleotide, right? So no matter what, if you're looking at an A, the sugar, or A's, T's, C's, or the G's, whatever, the sugar and the phosphate, they all look the same. It's the base that's different. The sugar and the phosphates, at least if we're talking about DNA, A, T, C, and G, all those sugars and phosphates are the same. If we were talking about RNA, where there's a U instead of a T, even then, for RNA, all the sugars and bases of the sugars and phosphates are the same. It's that the bases, the 4 bases that define it. And to remind you, and this is going to come back up later, when we talked about amino acids, right? There was 4 different parts. There was a carbon bonded to 4 different parts. And there was that one part. They were all the same. All 3 parts were the same, except that one part, which was the amino group. It was the amino group that was different. It defined one amino acid from the other. So same here. 3 parts are all the same. It's the nitrogenous base that's different. And that's the part that makes it unique, that makes it an A or a T or a C or a G or a U. So any questions about the nitrogen space? Okay, let's talk about the relatively unimportant for us, sugar. It has 5 carbon atoms. You don't need to know that. You could completely brain dump that and completely understand how things work. Of those 5 carbon atoms, 4 are in the ring, 1 extends above the ring. Again, you guys don't need to understand that. And also, the ring includes oxygen atoms. And again, you don't need to understand that. That's okay. So basically, every detail about sugar, you can just brain dump. You can understand everything I'm about to teach you without knowing this. Now, if we were really looking into it, this was a 100 level course, and this was a course for biology majors, then that information would be a little bit important, because when we really look at how things work, you would need those details, but we're not getting that deep into it. So, are there any questions about the sugar? Okay, let's talk about the phosphate group. Yep, let's put a big X to this. We'll talk about it. But again, this is information that you could just completely not understand, not remember, and completely understand what I'm about to teach you. So it's unimportant, in my opinion, for that reason. But yes, there's a phosphate at the center of it, or a phosphorus. That's where it gets the name phosphorus group. See right there is a phosphorus. This is where the nucleic acid gets its name. So we've already talked about the base, right? The nitrogenous base. Well, the phosphate group is the acidic part. That's where you get its name, where it gets its name. And then your book also points out that there's a negative charge on one of those oxygen atoms. Excuse me, one of those oxygen atoms. And again, none of that is important for you to understand the rest of the information in this chapter or this semester. So are there any questions about the phosphate group? Okay, before I move forward, obviously, let's take a step back and look at the big picture. You do need to know the 3 parts of the nucleotides. And again, you can just remember base, sugar, phosphate group if you want. But the most important thing, I guess, as far as it's understanding the upcoming information is the nitrogenous base. Make sure you understand the nitrogenous base is the part that sticks out, right? Because it's the sugar and the phosphate, they're covalently bonded to it themselves. It's the nitrogenous base that sticks out, and it's the nitrogenous base that defines which nucleotide it is. So if there's no questions, let's talk a little bit more about the fact that there's different versions of that nitrogenous base. For DNA, there are 4 different types of nitrogenous bases. We've already said this, I'm saying it again. There's A's and T's and C's and D's. I'm gonna put an X through this. This is important information, so I'm not putting an X through it like you don't need to know it, because you kind of need to understand this a little bit to help you understand what's coming up. But I'm putting an X through it, signifying that it's not going to be on the exam. But anyway, like I said, there's 4 different types, A's, T's, C's, and G's. And of those 2 or 4 different types, then they're subcategorized. They're actually lumped in together because the T's and the C's are single ring structures. So you can see right here, here's a C. That's what the C looks like. There's a T. Oops. Come on. There we go. There's the thymine. Single ring structures, right? The other 2, the A's and the G's, are double ring structures. And that will be slightly important later. And I'll just go ahead and give you the reason is, this is going to be a horrible drawing. If you're trying to put 2 strands of DNA together, and you have, I'm literally going to draw it as a ring, even though it's not circular. If you have a double ring structure here on this one, then you need a single ring structure here for it to fit. I drew that wrong, but basically, if you had a double ring here, and a double ring here, and then a double ring here, and a double ring here, it wouldn't fit. It'd be too big, right? So what I'm getting at here, and I'll talk about this in more detail later, is whenever you're looking at how they pair up, it's always going to have to be a single ring paired up with a double ring. And that's part of how the cellular machinery that you're going to learn about later and how it works. Because if it just doesn't fit, if it literally doesn't fit, then the machine kind of knows, oh, I made a mistake. I need to go back and fix it. But we'll talk more about that later. For now, just know that there are 2 types. You know, some of them are single ring, some of them are double ring. You don't need to know which is which, and none of that's going to be on the exam.
So only 3 can be in it. time course.
Correct, yeah. Only 3. 3 is, meaning that for those of you online who can't hear, see like this top one that I drew, that's a good one, right? I mean, it's a bad drawing, but that will fit because we have a double ring and a single ring. Another thing that wouldn't work would be a single ring and a single ring. It'd be too skinny. And again, the machine that puts those things together, the cellular machine would be like, oh, something's wrong, that didn't fit, I need to go back and fix it. Not that you're gonna get tested on that, but it's not all about the exams, right? I want you to understand how these things work. Any other questions? Good question. Okay. As I've already said, on this chapter and in chapter 3, RNA and DNA have the same chemical structure, except RNA does not have a T, it has a U. And you do need to know that. As a matter of fact, there's something that you won't understand yet, but I'm going to talk about it again later in this chapter. But there's going to be a point in this exam where I say, transcribe this. Meaning I'm going to give you a strand of DNA, A's, T's, and C's, and G's, whatever. And then you're going to have to make a strand of RNA from it. And remember, these are multiple choice exams. So if I tell you to transcribe something, that basically means I need you to write, I need you to make a strand of RNA. So in this multiple choice exam, when you have to transcribe something, if anything has a T in it, you can always just cross that out. No, that's not the right answer, right? Save yourself some time. If it has a T in it, and I'm asking you about a strand of RNA, you know it's not the right answer. Likewise, if for some reason I have anything that says, replicate this strand of DNA, meaning here's a strand of DNA, tell me what the other strand of DNA would be. If it has a U in it, you know that would not be the correct answer because a U would be an RNA, not DNA. So yes, that's not trivial for the exam. You need to know the difference between the two. Another thing worth noting that I am going to put next to it, because I'm not going to ask you, because you don't need to understand this to understand how this chapter works. But RNA has a different sugar than DNA, and that is actually the basis of their name. So it's ribose for ribose for RNA, and for DNA it's deoxyribose, hence the DNA and RNA. But again, you don't need to know that. You can completely brain dump that and understand what I'm about to teach you. The next word then, or I guess phrase for independent work, since I just said it, is brain dump. I know online, make sure you guys heard that online, brain dump. Like when you just completely forget something that you learned. All right, if you download the PowerPoint, you can watch this little animation.
So was there brain?
Speaking of which, you can watch it if you want. Yes.
Can you go back?
Yeah. But speaking about that animation, like I said, you can watch it if you want. But I should shortly share some good videos, some pretty good videos about this stuff. Usually from the same source, that Crash Course Biology with that guy named Hank. I just love his work. It's short, it's sweet, it's to the point, it's relatively entertaining, and it makes sense. You good? All right. So yeah, watch it if you want. You can also watch this too, the Hershey Chase experiment. I'm not even going to say what it is because that would be a good independent work question. That could be a 2-sentence answer, or you can write a whole paper on it, whatever you want. But what was the Hershey Chase experiment? Look it up. Or you could also watch the video to prime your information. Like, oh, okay, I get it now, and then you can just find your source to back up what you learned in the video, or maybe add some more details if you want. All right, now going back to the storytelling. So we're done with that bullet point. We're done with our little review, right? A lot of that was old information, the structure of RNA and DNA. Now we're going back to the storytelling of how we learn these things. And we're going to talk about Watson and Crick's discovery of the double helix. There's another good or decent independent work topic. It's a little bit off topic. It's about biology, sort of. It's about biologists. But between Watson and Crick and some other people I'm going to teach you who are really famous, let's just say one of them, I won't say has been canceled, but when you're dealing with people from back in the day, a lot of times things come to light that in today's society, you're like, well, what? You can't do that. You can't say that. So just say that about these people.
Do you hear me?
If you want. Anyway, specifically, the names of these people are James Watson and Francis Crick. You don't need to know their names. I'm not going to test you on that. I might have a video if I can find it. It's a decent video. It's not a biography. It's not a documentary. It's actually a movie about this. like a movie that's supposed to entertain you that came out in the movie theaters, but I'll see if I can find it for you. Anyway, these are the two people who determined that DNA is a double helix. Now, if you know this story, you're probably inside thinking, well, oh, there's more people. And we'll talk about those other people later. And they are very important, and it is very controversial as to who discovered what and how much, but that's not what this class is about. This chapter, we're actually just learning about DNA and RNA. It's not so much about who discovered it, even though we teach it. We're not, that's not the focus of this. But anyway, like I already said in the last major bullet point, DNA is a double helix. What is a double helix? It's a spiral with uniform diameter made of two polynucleotide strands. It's a really fancy way of saying, like you already know, there's two strands of nucleotides, right? There's a single strand of the A's, the T's, and the C's, and the G's that are covalently bonded to themselves by the sugar and the phosphate. Then there's another strand covalently bonded between the sugar and the phosphate. And you put those things together, so now you have two strands, and you twist it. That's what we mean by double helix. Double because of two strands, helix just because it's twisted. And honestly, and I'm not putting next to this for the exam because you do need to know that it's a double helix, but for understanding what I'm going to teach you later, really the helix part isn't even important. The fact that it's twisted for us and what I'm about to teach you and how I'm going to teach you, the fact that it's twisted means nothing. The fact that it's double, that means a lot. That will be important. You'll see why as we get into it. How did it all work? Big ol' X to this because, again, it's not the focus of this chapter. It's really good information if you are, slightly interesting information if you want to look it up for independent work. It is, like I said, a little bit controversial. But anyway, Watson saw an X-ray image of DNA that was produced by someone named Rosalind Franklin. That's where a lot of the controversy comes from. Like, did they steal her work? What definitely is not controversial is the fact that she didn't get enough credit for it. But anyway, yeah. He saw an X-ray of DNA that was produced by Rosalind Franklin, and then they got together, they used wire models, they constructed a double helix that conformed to everything they already knew about the DNA, right? So they knew a bunch of stuff, like I talked about earlier, like in the 50s, they knew, 30s, they knew this, 40s, they knew this, 50s, they knew this. And, they saw the picture of DNA, they put everything together that they knew, and then finally they were able to figure out the shape of it. And that's what it looks like right there. They figured it out. Another reason I really want to find this video for you guys, the reason I find it kind of interesting anyway, is like these two, who are like some of the most famous people in biology now, Watson and Crick, they weren't like great students. They were actually kind of lazy. They were just in England having a good old time. If I remember correctly, how do I not remember this? Only one of them was from England. The other one's American. Maybe it was Linus Pauling. He was the British one. Anyway, fact is that I find it interesting that they just weren't like brilliant students. They weren't geniuses. They weren't working hard. They would just like to drink at the pubs a lot, and eventually just stumbled into this. But again, that's not the focus of this chapter, so I'll move on despite how interested I am in that. So the model of DNA is like a ladder twisted into a spiral. Not that you need to write any of this down. I'm just trying to paint the picture for you. Like I've already said, it's a double helix, right? So think of two strands. This is what we have here. Two strands put together and then twisted, right? That's what we mean by a ladder twisted into a spiral. These sides right here, right? The sides of the ladder represent the sugar phosphate bonds. And those rungs that connect the two sides of the ladder, the sugar phosphate bonds, that represents the bases. And again, I'm not going to test you specifically on that. But it does help to understand how it works. And it's the base pairing rules that dictate which base on this side gets put with which base on this side. Talk more about that moving forward, but I've already hinted at that. Remember I said some are single-stranded? Some of the bases are single-ringed, some of them are double-ringed, and you need three, right? So if you have a one ring here, you need two rings here. If you have 2 and 2, that's too big. If you have 1 and 1, that's too small. And we'll come back to that. The next slide I'm going to show you is just a picture of a twisted ladder. But in the meantime, are there any questions about this? All right, here's what it looks like. There's your ladder. This time it's a rope ladder. That makes a lot more sense. That way we can twist it. Then you twist it, and then there you go. There's your ladder version of DNA. Here's a better version. Here's a little bit more accurate, right? All three of these pictures show the same thing. They're just different versions of it, right? Here's the ribbon model. Here's the atomic model. Here's the computer model. I'm not going to show you those pictures and say which is which. I'm not going to show you this picture and leave out the letters and tell you, which is which. That would be something for like a 400 level biology major. But yeah, so what is important that we're going to talk about later is the C's attached to the G's, the A's attached to the C's, right? And we're going to talk about that moving forward, and you do need to know that. But for now, I'm just going to leave it at that because it's just an introduction. Any questions about that picture? All right, let's move forward. You download the PowerPoint. There's this video you can watch here. Again, I'm going to hopefully show better videos for you. Let's get back to the story. In 1953, Watson and Crick published their paper that proposed the molecular model for DNA, right? So all that stuff I told you, they saw the picture of the X-ray. There was some other stuff I didn't even mention that they had figured out from teammates. They put it all together. They made a wire model of it. They wrote a paper about it. And then in 1962, so nearly a decade later, they actually received the Nobel Prize for it. Also a guy named Wilkins. And you can look that up too. Who was Wilkins? For independent work. And I do like that Alicia Book pointed out that Franklin should have deserved a share of that prize, but Franklin was dead at that point, and they don't do that, with the Nobel Prize. Anyway, if you click this picture of the Nobel, like if you download the PowerPoint and click that picture of the Nobel Prize, I think it brings you to a video. I really don't remember, honestly. It's been so long since I've made it and since I clicked it, but if you're curious, give it a click and see what happens. So, any questions about this slide? Gee, let me put a big old X through this. This is just telling you the story. None of this is going to be on the exam. This is not a history of biology class. This is a biology class. All right. So Watson and Crick wrote in this paper that they won the Nobel Prize for that the structure immediately suggests a possible copying mechanism for the genetic material. So in your brain, back up, to that earlier slide where I showed you, I'm trying to remember exactly what it said. Basically that until they figured out the structure, they didn't know how it copied. They didn't know how it went from DNA to RNA to protein. They didn't know how the information was transferred from one generation to the next. But as soon as they figured out the structure, and hopefully this is going to come to you when I show you, it immediately suggested a possible copying mechanism for the genetic material. So as soon as they figured out the structure, they figured out how it worked, essentially. Because it is the structure of the DNA that points towards the molecular explanation for life's unique properties of reproduction and inheritance. Right? So again, it's a pretty big deal here. Because as you learned in chapter 1, there are those 8 properties of life, the things that make us alive. And this is the scientific study of life. This is a big deal. You're learning about one of the things that makes all life on Earth possible. As boring as it may be, it is really, really important. Anyway, obviously, I'm not going to read all this, but this is just a little excerpt from their actual paper that they published. Any questions about this slide? We might be able to time this perfectly and finish the first main bullet point today. So anyway, the next thing we're talking about is DNA replication. And you know a little bit about it so far because of mitosis. What do you know about it right now? You know that it happens in S phase of interphase. When a cell reproduces, it has to duplicate its information, right? It has to provide a copy to its new offspring, but it also has to keep a copy for itself. Because remember, the cells split into 2 in mitosis, right? So it's not like the cells, like one cell went off and died and the other ones went off and lived, right? No, they both split into 2. And they're 2 basically new cells. So again, going back to what we talked about a second ago, it was Watson and Crick's model that suggests that each strand serves as the template to guide the production of another strand. This will make more sense when I show you. Here's what I'm saying. Here's the original strand of DNA. And once you go through S phase of interphase, that thing opens up, and then you use that opened up version as the template to make a new strand. So what you're left with, and this will be a question on the study guide and probably the exam too, what do the new DNA helixes, what do they consist of? Consist of an old strand and a new strand, right? Each one of them consists of an old strand and a new strand. And let's go back to what I told you about the structure and function thing, even though the words are on here on the board, but let's talk about it. It's a covalent bond It's the strongest bond, right? It's a covalent bond that keeps this strand together, right? The sugars and the phosphates that make that strand a strand are covalent bonds. Same here. This strand, it's a covalent bond that's keeping those sugars and the phosphates together. So, does anybody remember what these little dashes mean? Not that I expect you to. I never even asked you to begin with. We talked about it, but I never asked you this question. I never even talked about it much. But these little strands represent hydrogen bonds. So we have one strand that's covalently bonded to itself, right? That's strongest of the bond. We have another strand that's covalently bonded. That's keeping it together. So what's keeping this strong bond, this strong strand together with this strong strand? It's the hydrogen bonds. And remember, hydrogen bonds are the weakest of the bonds. So again, once they figured out this structure, they also knew that that would be hydrogen bonded to this, and that would be hydrogen bonded to that. Once they figured that structure out, they knew right away that must be how it works. You could just picture, if I were to pull this thing, it would just unravel, right? And the single strand of DNA is going to be fine because that's a covalent bond. It's the strongest. But these weak little hydrogen bonds aren't going to hold up. So I'm basically going to be able to unzip this, right? And they knew right away when they saw this, like that's what must happen. You can just unstrip it or unzip it and then use it as a template. And that's what's going on here and what we're going to talk about in more detail later. Here's a C, right? So it goes with a G. What you can't see here is there's like a cellular machine that's clamped on and is reading it. It reads an A. And it's like, okay, I need a T. And it brings in a T. And a T gets covalently bonded here, which then hydrogen bonds to that A. And then this one's a G, right? So the cellular machinery says, okay, I need a G. So it covalently bonds it to this strand, and then that gets hydrogen bonded to that strand. That's how it works. Basically, it unzips it, and there's a cellular machine that just goes in and reads the code and builds the complementary strand based on what needs to be there. The A's go with T's, and the C's go with G's. Download this PowerPoint. You can watch it in action. It's a really cool video, I think, because it actually shows what the molecules look like. It's hard to see right now, but yeah, that's the cellular machine, so to speak, and it's clamped on this strand of DNA. And you can see these nucleotides floating around, but then it grabs them and attaches them to the new strand of DNA. It's really fascinating stuff. Also, you can watch this version, which is easier to understand and better for learning because you can actually see, okay, it represents it, right? Like there's a strand of DNA, there's a strand of DNA. It's a lot easier to see and understand. It's not as cool because, that's what it actually looks like, but this will help you learn it a little bit better. So watch that if you want to download the PowerPoint. Here's another video you can watch. If you watch this one, make sure you don't pay attention to the words because there's a lot of words that I don't even teach you. Your book doesn't teach you. Lagging strand versus leading strand, template strand, primase, all that stuff you don't need to worry about. But basically, there's a cellular machine. Really, it's a bunch of machines working together. So you've got this thing that unzips the DNA, right? Because here's a double strand of DNA. It unzips it into two single strands. Then you've got this machine that clamps on that I was talking about earlier that reads the code and builds the new strand of DNA. It's really fascinating stuff. So what is that machine called? That is what you need to know for the exam. That will definitely be a test question. What is it? What enzyme, what cellular machine builds DNA? What is it that makes DNA? The answer is DNA polymerase. You need to know that. We teach it as one enzyme, but really, like I said, it's a bunch of things working together. But you need to know that. It is DNA polymerase that makes the bonds between nucleotides of the new DNA strand. In other words, how do we make new DNA? With this enzyme called DNA polymerase. This stuff here is what I've already mentioned, except now you can see it in writing. The incoming base pairs with the complement on the template. So remember, it unzips it, it reads the template. It's like, all right, here's a T, I'm going to bring in an A. Here's a C, I'm going to bring in a G. Here's a G, I'm going to bring in a C, right? That's the template. The template tells it which one to bring in. Your book also points this out, even though I'm not going to ask you. It is very fast and very accurate. How fast and how accurate? Well, it does 50 nucleotides a second. So think about that. 50 nucleotides, 50 nucleotides, 50 nucleotides, 50 nucleotides. That's how fast it is. How often does it make a mistake? 1 in a billion. 1 in a billion. Of course, to be clear, it makes mistakes more often than that, but it has a mechanism to fix its mistakes. So 1 in a billion times, it makes a mistake, it doesn't fix it. Speaking of that mechanism that helps it fix itself, it also helps repair DNA that has been damaged, well, just by anything, right? Your book lists some ways it's been damaged. It doesn't matter. When your DNA gets damaged, this thing, DNA polymerase, also has a mechanism to fix it, which makes sense, right? If you have a double strand of DNA and you just lost some nucleotides, and this thing's job is to put in nucleotides, they can just attach and put into nucleotides. When it doesn't do that correctly, though, that's when we get mutations. And we'll talk about that later in the chapter. We've already seen this picture. Like I said, it reads the strands. There's an A, bring in a G. Here's a C, bring in a G. Here's a G, bring in a C. Right? It reads the strand, reads the template strand, and brings in the new one. Here's a little bit of a review for you. As I've already said, DNA replication happens in the S phase of interphase. You still need to know that. Here's the new information. Where does this happen? In a place called the origins of replication. This will be important because later in this chapter, you're going to learn about something called transcription. And one of the questions is going to be, where does transcription start? Then you're going to learn about translation. And one of the questions is going to be, where does translation start? So for replication, transcription, and translation, there will be a question that says, where does it start? So the answer for this one is, DNA replication begins at sites called origins of replication. Notice the origins is plural. That's because it doesn't start on one end of the chromosome and work its way to the other. No, there's multiple strands and multiple starting spots throughout the chromosome. They just kind of work their way out until they meet each other. And that creates something called replication bubbles, but I'm not going to ask you about that, so I'll go ahead and put an X through that bullet point. But that's a great word for attendance, bubbles. Also a funny character on a show called Trailer Park Boys. It's Canadian, it's funny. Any questions about this slide? You download the PowerPoint, you can watch this, which shows it in action. It's an animation of what I just taught you. And that one too also shows it in action. And that one too also shows it in action. And we did perfect because it's 8:50. So when we get back on Monday, we will start the second major bullet point of this chapter. And neither one of you are wearing red, so we're done for the day. If you guys have any questions, I'll be in my office and online for office hours. Have a great weekend. Don't forget to do your labs.